Right. So I can do more samples. I can do that much cheaper in New Zealand than I can at Neogen. I basically can do 2 animals for the cost of 1 there.
Okay.
Sure. Okay. All right. That's helpful there. And as I said, we're not there yet, but it was just, is there a way we could actually practically test more animals? Because if we can't really, we can't proceed. So without solving that problem first.
So,
Okay.
Wait.
And let me
Yeah, go ahead, Dr. Murdoch.
Okay, so let me just say, so even if you were to go with Neogen, like what you could send your stuff in, they don't do the analysis either, right? So really what they can sell you the genotyping, they can give you the genotypes. You still need somebody to process that information for you. The NSIP takes that data and rolls it into eventually genomically enhanced estimated breeding values. That's what NSIP does, and they use the Neogen chip in part. We'll just say there's a bit of a relationship there. Anyways, but Neogen itself, they don't do the analysis. They would be almost like what Carissa will do for Flock 54. They'll sell you the genotyping, they'll give you some information back, and then it's up to you. I'd say Neogen does even less than what Flock 54 does. But they are a service provider. They can sell you the genotyping. You don't always have to use the same chip, although what we would do is look for overlap of the markers that Neogen has and the markers that that they sell at in New Zealand. There's a fairly big overlap, so we do that a lot on different projects. But if you want that other research and that other relationship information, PCA stuff, there's not a service provider that does that. That has to kind of usually come back to somebody like me or somebody else. I'm not the only one that knows how to do that, but people like that to do it. Then none of those service providers actually do that.
Okay.
First, we'll do the parentage. Criswell will do the parentage, and we've showed them how to do that kind of stuff, so they can do, and we'll give you back that kind of stuff. Yeah, and then the Neogen, that is more. They will give you back some stuff, but for the most part, it's meant to feed into their genomically enhanced DBPs.
So let's pretend that you would be willing to do that for us. What might that look like, as far as like a process or cost or any sort of, just so as we're thinking about it, we know what's realistic for everyone involved.
Yeah. So, I'm a university researcher, so I don't really have, a price. So the project that we did before, I'd written a grant to get funds to be able to do that for you guys. So moving forward, the genotype, I can't pay for the genotyping costs anymore, but we've already done most of the work. So we already have the process and the pipeline laid out. So paying for samples, getting them shipped there, it doesn't cost us a whole lot to, re-push the button with new stuff in, new data sets in there. So we're happy to do that for you. Of course, we always try and think about, we want to give that kind of, information back to producers, but then think about how can we use that to investigate and answer research questions. That's the benefit to me, right? So if I do it for people, there's no charge, but then can I then ask a research question like the one Dr. Sponenberg is proposing, right? How much change has been, right? So then can we then do those analyses? As we engage a student to do those analyses, they do the analyses, they learn how to do it, they write a paper, it goes out in the scientific domain. That's their benefit. And then we train somebody into doing that. So for me, that's where my payback is then I potentially can use that data for other research questions.
Okay.
Okay.
So it sounds like if we could get the folks who actually want to have their sheep tested to just cover those hard costs, the supplies and the shipping and that sort of stuff, that the actual analysis could possibly be done for free, with the understanding that the data would then be available for future research projects. Okay.
Yep. Yeah. And then the other thing that I just want to add in there, though, is like for a situation like this, probably what we would do is we would wait until we get sufficient numbers, right? So if somebody sends me one TSU for one sheep, you might wait, right? So is it worth redoing all of the analysis? Is it worth shipping all of these and doing, sending the sample to New Zealand for one sheep? No, we probably want to wait until we get a few more, then we can push the button again and see how those few more sort of pan out and give you the same kind of output that you have gotten. But yeah, if somebody sends me a sample and then wants their analysis by next week, yeah, that probably just isn't going to happen.
Okay. All right.
Sure, we'll be careful to set expectations there if we go in this direction anyway.
Yeah, yeah. I mean, if you're sending your stuff to Flock 54 and you want your scrapie and you want all that kind of stuff, then they'll have their turnaround time, and they will, because they are a service provider, right? So they will give that back to you. But yeah, for the bigger thing and for research, it's just a little different.
To take a little bit of a divergence, we talked about scrapie a little bit, and I'm actually the person that imported Wensleydale sheep into the United States in the 1980s. And I am also the person who had the only case of scrapie in the Wensleydale breed ever. And scrapie resistance really makes me nervous, because I think what we're doing, I don't think it's resistance. I think it's a prolonged incubation period. And I really do advise caution on which direction to go with that. The Icelandic approach has been, don't necessarily breed for resistance, just select susceptible sheep for absence of the agent. And that actually may be a little bit better. That's my own little soapbox, and there you go.
Yeah, I agree with people use the term resistance, but I think it's reduced susceptibility. And that there's actually another marker that's on the Flock 54, which is for atypical scrapie. Those are animals that by other means have been considered resistant. Again, I also agree that the word resistant should not be used, because if you expose that animal with enough infectious agent, I'm not sure there's any genetics that will avert them from getting that disease. But yeah, I think we give that just sort of, I guess, genetic predisposition information, and we can phrase it that way. I'm not really the person that phrased it susceptibility and resistance. I just going with the terminology, but I totally understand and agree with you. That some people say and have the same perspective as you, that selecting for genotypes with lower susceptibility might just be prolonged incubation and might just lead to more of atypical type scrapie cases with that infectivity, if in the presence of infectivity.
Okay. But I say we'll manage expectations as best we can there. The only things I have remaining are a couple of questions that we've already been asked, and these are for both Dr. Murdoch and Dr. Sponenberg. If any of the sheep that we tested, even though they were registered, if they were crossbred, as long as it was a recent crossbreeding, that would have taken that dot out of the cluster. Is that correct? And that's how I understood it, but I want to be sure before I answer someone who's asking this question.
So, okay, say again.
If one of the 78 were crossbred, their their PCA representation would have fallen outside of, or probably would have fallen outside of, the grouping of purebred sheep.
So usually you will see those dots a little bit more distant, but the when and really what that dot really the grouping tells us how similar and how close those genetics are, right?
Okay.
So a dot that's outside of that just means that it's not genetically as similar to all of the other ones. That may or may not be a result of crossbreeding, right? Like if you bring in, I would suspect if we were to sample sheep in the Caribbean, they are going to be a little bit different, right? That doesn't necessarily mean that they're not St. Croix. It means that they are going to be a little bit genetically different to what that breed population is here. Again, that doesn't mean everything here has been crossbred or outbred. So I think we have to be a little bit careful on how we interpret that. It could be that all of those sheep are crossbred. They're just crossbred in a very similar way, right? So they're genetically similar, right? That's really all that's telling us is how genetically similar they are to each other, which is why in the beginning I said if we can sample as broadly as you think the breed is, then we capture that circle. And I don't yet, that's a question for you guys. How well did we capture everything? There may be some that we failed to capture in that might be a little on the outskirts. I think when you think about those PCA plots, if you see a dot that's now boom in the Katahdin group or boom in one of the other, then oh, okay, that animal is more genetically similar to those groups than it is to your group. And I think that's as far as I would go on that.
Yeah, I think that was kind of the question. So, like, say if I have animals in my pasture who are half Katahdin, half St. Croix. If I were to have slipped one of those samples in, would it have been fairly obvious on the PCA plot? Like, would that animal have been like, whoa, way up towards the Katahdins? I think
it should be, it'll be closer to the Katahdins.
Okay, that's what I thought.
Okay. That was what we thought, but I said, I'm trying to stop people from making huge leaps based on just an impression of data that doesn't really support that. The next question had to do with the Flock 54 results. When I look at the sheep, their susceptibility to various diseases seems to be, mostly wild type, not susceptible. Are there any normative databases for all sheep? Can we legitimately say that St. Croix have more genetic resistance to more diseases than other breeds? Or are all sheep like that? In other words, they're pretty average. The St. Croix breed is pretty average compared to other breeds.
Yeah, that's a good question. So those sheep disease reports, we get them from a public database. It's called OMIA, where they report different diseases. Some of those diseases are going to be more common in certain breeds that are not St. Croix. So the frequency in which you see that might be much more rare in any breed outside of the breed that it was tested in. So those so a lot of those are, I would say, rare to moderately rare genetic things that we see out there. For example, spider lamb, right? That's mostly a Suffolk thing, right? So the fact that you don't have any, I guess that's not really a surprise, right? If we were to look in other breeds of sheep, like, I don't know, Rambouillet, you would probably not going to see any of those alleles in there either. Yeah. So I don't know that it means that they're better than other sheep or Yeah, I would just say that those are the causative mutations that we know in all breeds of sheep. And then just it's an output of and a part of those genetic analyses. So we would for sure look at them, although you have to remember that some of them are probably not that relevant. So not all of them were diseases. Some of them are diseases. Some of them were increased fecundity. One of them
Right.
is myostatin, so increased meat, right? Which we only usually see in Texel type. So I mean, we just test everything and give everything back. So and there is kind of little bits of information. I don't know how well they go into it, but yeah. So I think, we just try and look at everything on a very broad, and some things are going to be applicable to some breeds and not really to other breeds. But, it's still always a good thing never to see any of these. So even if it hadn't been reported before, that you're still not, don't have alleles that are susceptible for that.
Okay. Okay. So it sounds like saying, St. Croix are genetically resistant to more conditions or diseases than other sheep really isn't a fair statement to make,
No.
is what it sounds like.
Yeah.
So, okay. And that's exactly what I'm trying to guard against. It's like, oh, wow, making these kind of statements that aren't really supported. So, the last question I had was about color. I had asked you before about whether the PCA shows us that St. Croix with color do or don't form a particular subgroup, I guess, within the genetics. And you said that's not really what the PCA is looking at. And I'm beginning to take that in. You mentioned an association study. Is that something that can be done with 78 or 92 samples? Is it ridiculously expensive? Is it something that can't be done? Because color is a huge issue, not for us, but for the separation of two different registries. And it just seems like if we had an answer to that, it would be helpful to us, but I don't know if that's even feasible.
So the markers on those genetic tests, a lot of them are color markers, right? So we know a lot of genes that make whiteness, and we know genes that make darkness, like MC1Rs that makes a darker coat is that same gene in cattle and any mammalian. So there's a lot of genes that are on those panels. With the bigger test that you have, we could do the association study. It's really just running it through a different program. It's not really a PCA thing. But for that, we need to know, we need to have color information on all of those sheep, right? Mm-hmm. So, and then the other thing to say, when I run a genome-wide association study, you also are trying to, think, you have to kind of think about it a little bit differently, particularly with a small population. So for example, if all your sheep are all white and they're all homozygous white, when I run it, if I never have anything dark to see it differentiate against, I might not be able to see that with the small population. So we have a couple different ways of doing things. We do these single nucleotide GWAS things, or we can do these runs of homozygosity where we say, okay, they're all white, and then, oh, here's the KIT gene in here, and they're all the same. So there's a couple different ways. Of course, all of these analyses are always better when you have more animals. So more animals, and when you have variation in the trait that you're testing for, it's much easier to do that with, right? And then the 60K tool is better to do that. The Flock 54 there's a little bit of that in there, but it's not really meant for that. The Flock 54 is meant for when we know those associations, and have proven it, then we can put it on those lower platforms.
Okay, Dr. Sponenberg?
a brief aside, and this is for Dr. Murdoch especially. Most of the color variation in the St. Croix is going to be at the agouti signaling protein locus. Unfortunately, and I have no idea on the 60K what all that's going to pick up because it's a really, really complicated locus. But the MC1R should almost be invariable. Unfortunately, KIT probably, there may be some, but that's not actually going to contribute to the whiteness. So it's, of course, for me, it's a fascinating question, but I'm not sure the answer is going to be all that easy.
Yeah, so the agouti marker is on flock 54. I did put the agouti marker on it, but there's just one mutation on there, right? And so as you know, so we put one that was previously reported in the literature that may or may not be what you guys are seeing. And the agouti marker is, again, it's not, it could actually be methylation and not a nucleotide variance in there. But if we know that, we have ways that we can actually go through and sequence that region if we know that's what we're looking for. So there are, I think, one or two agouti markers on both those platforms. Whether that variant would associate, I'd have to test.
And one of the, I think actually the underlying question is, if they identify the colored sheep and they identify the white sheep, is it possible to figure out if the colored sheep overlap perfectly with the rest of the genome of the white sheep, or is there evidence of introgression from something else? And that it gets to be complicated because some Barbados blackbelly sheep are actually blackbelly St. Croix sheep because of the way they were brought into the country. So complicated question, but a little bit different. Not necessarily looking for the genetic basis for the color, but whether the sheep that are colored are legitimate members of the breed, if that's correct.
That's exactly the practical application that somebody is going to want to make.
Yeah,
I know that for sure. Okay, I do know for sure that's at least some color sheep are in the sample. I don't know how many. I believe 6 at a minimum, but I'm not sure that is enough to be helpful. And the color ranges from, a couple spots of light tan to some majority dark black color of a sheep. So it's all over the genetic cause map. The color it's not all uniform either. Okay, that bears more talking about within our group, whether we even want to go in that direction at all. But thank you for the input. So we're not making, we're not trying to find our way in the dark about what we could or couldn't reasonably expect to learn. So Crystal, that was all I had. Dr. Murdoch, Dr. Sponenberg, if you have anything to add, or Crystal, you too, just please throw it in. But that's the end of my list of questions. So.
Well, the other thing I would add is, along with color, we also have the issue of skurs.
Yes, that's true.
And horns, which horns are very rare, but they do occur. And neither of the groups allow those to be registered, but I think it would be a fascinating project at some point to go, so that, they're horned, but they're still obviously a St. Croix via their genetic study. Anyway.
Yeah, so we do have the horned locust both on flock 54, but also on that 50K. So you want to know sex, and then so what's important for any of these studies, whether it's a GWAS or whether it's a run a homozygosity, where we're looking at various introgressions and heterozygosity regions. What's important is to have all that other data, right? So what color is the sheep? Do they have skurs? Do they have horns? And once we collect enough of that kind of data, then we can start to analyze this, right? So, I mean, the horned locust in sheep has been published. It's out there. It's nothing new to discover. So we can certainly test if animals are are heterozygous, right? Because it's a homozygous recessive trait, I believe, if memory serves me. Yeah. So then if you're starting to get that allele in there, if animals are carriers and things like that, you can ask those kind of questions. But yeah. What happens is we need that call it phenotypic data, whether it's coat color or skurs or horns or any of those things that your breed association is interested in monitoring. We want to collect that data for those animals with their genetics. And then once we get more samples, then we can do those more definitive those definitive analysis in order to help identify that.
So it sounds like
Okay.
if we continue on this project with collecting more samples, we need to have a little bit better data collection method instead of our handwriting chicken scratch one that we used before.
Yeah, and I think, you know, as a breed, what do you guys think is important? So if
Right.
you say horned and skurs and color, then that becomes a piece of information everybody needs to fill out, right? So
Mhm.
people will, you know, they'll put something here in this phenotypic form and somebody else puts something else and
Yeah.
they miss this other data. But
Yeah.
if that's what
That's exactly.
if that's what you guys want, right, then we want that information filled out by all producers. And then when we run these types of analyses, and we can run them on a whatever quarter basis or whatever, and get information back, or on a yearly after lambing kind of cycle, and get information back to people. But yeah, without all of that other kind of information, it's really, we're 4 nucleotides that we're trying to make sense out of, right? So.
Right. Yeah. Okay.
Okay.
And then
Dr. Sponenberg.
sorry, I had one more question, and it goes back to that cost. What is the hard cost for that test? Because I know we would not want our members to cover the actual sending of the stuff over to New Zealand. What's the hard cost for each of those? Just so we have that in our minds.
So I pay $17 a sample for the research test, right?
Okay.
Then I pay to ship that over.
Okay.
So whatever that shipment fee is, I don't know, like $50, and that's $50 if it's one sample. That's $50 if it's 200 samples
Right.
or 2,000 samples.
And the TSU, which we would need. Yeah. Okay. Thank you.
Yeah.
All right. Now I'm done.
Okay. Dr. Sponenberg, did you have any questions or anything to add to that? And we're looking to you for help on, you know, what's important, what might be useful for purposes of breed conservation. So we'll get to that. We don't have to do it all today, but that was, you know, we don't know. We're just reaching out and thinking and brainstorming ideas at this point.
I took some notes, and I'll write something up and then send it to the three of you, and then you can mull it over and see, because there may be some disconnect on which questions people actually really want answered. I may be able to help navigate that or help translate that. So nothing right now, but I'm not quitting yet either.
Okay.
Well, we appreciate that. So, okay, I think that's everything then. And again, thank you both for participating.
Yes.
There, we're very excited about this, but you know, we have the sheep in our pastures,
Yeah.
So it's easy for us to be excited, and that's why I appreciate your involvement too. So, all right, unless there's more, I think we're all good.
Okay.
All set? All
Thank
right, we'll
you.
be in touch about a February date also.
Yeah.
Just as a
Wonderful.
closing. So, all right,
Okay.
thank you all very much.
Yep, thank you.
Bye.
You're welcome. Bye-bye.